Welcome to Flora and Friends, your botanical cup of tea, a podcast for plant lovers of any kind. We welcome guests to our botanical tea break to explore the history, science and meaning of plants for our lives. My name is Judith Lundberg-Felten. I'm a plant scientist, university researcher and founder of Flora-L Design, and I'm the hostess of your botanical cup of tea. Welcome to this second episode of the Flora and Friends podcast mini-series on Pelargonium plants. Today I'm going to take you on a little journey to South Africa, where we are going to visit Professor Nox Makunga's lab. And Nox is going to talk about the use of pelargoniums as a source of phytopharmaceuticals, so pharmaceuticals from plants. And she's going to highlight a few aspects of how they are growing and working with them in the lab, but also how this lab knowledge has been helping to inspire cultivation practices that can be helping for conserving the natural occurring pelargoniums outside of the lab. So Nox is a professor and a plant molecular biologist working at Stellenbosch University, and she has worked herself with Pelargonium species. And I'm very happy to have had an interview with her. It was very pleasant and captivating to get insights into the work and also to see how important this scientific work is for industrial purposes and for nature conservation. So with this, I lead over into the interview and I say, welcome to Nox Makunga.
Thank you. It's lovely to be here.
So can you tell us a little bit about pelargoniums in South Africa and their medical use?
Pelargoniums are very much integral to South African life. We have a large biodiversity of pelargoniums, and there are some medicinal pelargoniums that have even found themselves into the commercialized space. But they've been utilized by people for centuries for a variety of different ailments, and especially for respiratory infections and other treatments. And they're quite a wide range of treatments. It ranges from dysentery to diarrhea to all kinds of ailments. So they are quite explored locally for their chemical diversity and have been part of South Africans and their health for centuries.
Mm-hmm. Do you know something how people have discovered like that they had a medical benefit that one could use them for?
I think it's quite hard to trace how people find out about medicinal plants, especially if you go back in time. But with regards to Pelargonium sidoides, which is probably one of South Africa's most important phytopharmaceutics, it is said that there was an interaction with a Basotho healer and a European settler. And then this resulted in a transfer of knowledge with regards to the pelargoniums. And ultimately that led to the Pelargonium sidoides actually being taken to Europe, and it was made into a consumption cure. And that was probably in the 1800s. And that was the first time that South African medicinal pelargoniums found a commercial interest.
Mm-hmm. There's about 280 species of pelargoniums, if I'm right. Is Pelargonium sidoides the one that is most explored, or are there other pelargoniums that are used for medical purpose?
There are other pelargoniums that are used for medicinal purposes. So the sister species, Pelargonium reniforme, looks very much like Pelargonium sidoides. And so both of those species are actually exploited for medical purposes. But you also have Pelargonium graveolens that is actually exploited in the aromatherapy industry for its essential oil. And when people refer to rose geranium oil, that's the species that they're actually, referring to. So pelargoniums are exploited for the fact that they make these aromatic essential oils, but they have also these tubers that will have a concentration of medicinally important compounds. So, and those three that I've mentioned are not the only ones that are exploited, but are definitely, the three that have been commercially exploited. In terms of traditional medicines, a wider range of pelargoniums are actually exploited. And these are generally harvested from the wild, and local herbalists and traditional healers and naturopathic doctors will actually utilize that biodiversity. And it's quite a wide spectrum of different types of pelargoniums that are actually exploited in the traditional medicines markets.
Mm-hmm. That's very interesting. And as you say, for some of them, it's more the essential oils. For others, it's more the phenolic compounds. Is there a relation between those compounds and from which tissue they are extracted? For the sidoides, you said it's the tubers or the rhizomes. And for other compounds, are these other parts of the plant?
So for the essential oils, it's generally the leaf material that would then be placed in a hydrodistillation system to release that essential oil. And the floral parts, as far as I'm concerned, are not necessarily exploited for medicinal purposes, but they are edible. So they're actually quite fun to place in food and for decoration. So that's an interesting use, I would say. So it's mainly the leaves and then the underground parts that are exploited for health purposes.
Mm-hmm. And as you say, there's a lot of people that take this from the wild. Has this become an issue for pelargoniums in the natural context in South Africa?
It is a little bit of an issue because there are certain regions where Pelargonium sidoides is actually found, where populations have been decimated by overexploitation. And as a result, there are now farming practices that are also being used to bolster the biomass of material that actually goes into the formalized botanical medicines trade. We are quite aware that if we don't look after the plants in the wild, that we will lose the biodiversity. And so there are many efforts related to conservation and utilizing cultivation as a practice to try and provide alternative sources of having access to these metabolites that are of medicinal value. It has been a problem, but it's a problem that we are well aware of. And I think the industries that have commodified Pelargonium species are trying by all means to become a bit more sustainable in their practice.
Mm-hmm. So that makes us a little bit move into your research as well, because I mean, seeds from the Pelargonium sidoides, they don't germinate so well, if I know right.
Mm-hmm.
And so if you wanted to move them into a cultivation, what kind of possibilities are there? I know you have worked with some possibilities yourself. So if you can give us a little insight into that.
There are ways in which to induce germinations with pelargoniums. And I think one of my favorite ways actually, and this induces germination in a variety of different plants, is to apply smoke. Because the smoke contains a group of compounds that are called butanolides. And a set of Australian researchers actually named these after they characterized them and referred to them as carricins. And those carricins are able to induce germination in a wide range of species, including the pelargoniums. So I think it's a wonderful way to encourage germination.
Mm-hmm.
You can get smoke, water as well, and even filter papers that can be purchased that have actually been dipped in that smoke that still contain those compounds. And then you place the seeds, add a bit of water to that dry filter paper with the smoke, and then that will actually encourage germination.
Okay. That's very interesting. I had never heard about that. You could encourage germination with smoke. So if you have a barbecue at home, can you use that? You don't want to roast the seeds, I guess.
Yes, absolutely. No, you don't want to roast the seeds, but it does work very well if you have a barbecue. And it's pretty much any organic matter that comes from plants that actually that will release these compounds, the smoke-inducing compounds. And so a barbecue is just perfect. You can just have those seeds whilst you are setting up your barbecue, and then they will absorb that smoke, and then that will induce germination. And the reason for this is because a lot of the plants that are actually found in the Cape have evolved in fire-prone environments. And so the fire and the smoke are actually critical for their life history.
Mm.
And many plants from this area here in the Cape Floristic region only germinate once they've actually received a smoke cue.
That's very interesting, because I think that wildfires are part of ecology. Is not so much. No, we're not so much aware of this, because we often think about fires as well. Then the fire takes over, and then everything burns down. So obviously has been a problem. But that fires are part of the landscape and form the landscape is a very important part.
Yes, and that is what people sometimes exploit when you have recalcitrant seeds that are very difficult to germinate. Then you can actually use that as a system to induce germination.
Mm. So when you want to work with Pelargonium sidoides, and let's say you can work with seeds, can you work with cuttings, like make clones from them?
Yes.
Mm-hmm.
So a lot of the commercial farming that is associated with Pelargonium sidoides, they use a vegetative system where they actually replant the cuttings that they would have generated from their previous crop. And the tubers can also be broken and replanted, and then they will also induce a new set of plant growth. So all of those tissue types are quite amenable actually to regeneration and the production of new plantlets.
Mm-hmm. Which means that is it's a good way of being able to farm this and not to have to collect them from the wild and to destroy their ecosystem. And you have been working with a project as well where you have produced something that is called hairy root systems. Can you tell us a bit more about that?
So these hairy root systems are very interesting because it is stemming from a natural genetic engineering system where agrobacterium, which is a soil bacterium, and particularly the species rhizogenes, it transfers a segment of its own DNA into the plant genome. And once that's taken place, the genes that are actually sitting that came from the bacterium are then expressed by the plant, and that induces a set of plant hormones, and particularly auxins. These auxins often are linked to the production of roots in plants, and so the plant then manufactures this masses of roots, and those roots are an incredible biochemical factory that is able to generate an increased output of metabolites. And so we exploited that hairy root system as a means to source the key compounds, the phenolic acids, the coumarins that are produced by Pelargonium through utilizing that gene transfer system. And we are able to set these up in a tissue culture system where they regenerate through different cycles of time. And all they need is a medium that doesn't actually have any hormones because they're manufacturing their own hormones. And they actually have a very fast growth rate. So it's a wonderful system to be able to exploit for industrial manufacture of the metabolites that actually come from Pelargonium. And that is one of the reasons that we thought that that might be an interesting way to produce those metabolites. And lastly, it allows us in our laboratory environments to be able to actually study specialized metabolism, because those pathways that lead to the production of these metabolites are not necessarily very well understood.
Mm-hmm. So you have been able to set up a little biofactory with this system.
Yes, indeed.
Since you did this study, has there been any like commercial utilization of the system?
So the system hasn't necessarily been commercially exploited, and I suppose one of the reasons for this is because it is a genetic modification technology, and so there's issues that are linked to regulation of using GM-related products for medicinal purposes. But it provides us with an excellent scientific tool that allows us to be able to study metabolism, which then provides us with more fundamental information about how we can actually exploit these different biosynthetic pathways. I say this, that it hasn't been exploited on a commercial basis, but we also developed another system in our laboratories, and this was a non-GM system where we actually utilize a tissue culture production of Pelargonium sidoides. And that particular system is exploitable for the commercial industries. And we are at the stage actually of negotiating with some of the manufacturers here in South Africa for them to utilize a plant tissue culture based system as part of their agricultural setup. So that particular system that's linked to the tissue culture is actually more easily exploitable, and it directly links to the production of plant-based pharmaceutics.
Mm. Can you explain how you use that tissue culture system? How, if you use the tubers there, or if you use cuttings, and how can you favor the development of the root system and the tubers in that system? Because I think you are more interested in stimulating the below ground tissue as compared to the above ground for this commercial purpose.
So we devised a protocol or a regime to be able to do tissue culture, and it's quite interesting that you can set up all kinds of different cultures. You can set up root cultures, you can set up plantlets, you can even set up a set of cultures that are just a disorganized mass of material that's known as callus. And these different production systems actually all have the capacity to produce the metabolites that are monitored by industry. So that was a very interesting finding. And what was even more interesting was that when we looked at the extracts of these different Pelargonium plantlet cultures or even callus cultures, they were sometimes more active in the bioassays that we tested than the plants that would have been growing in the greenhouse. So there was something in that microenvironment that was actually switching on metabolism and favoring the production of some of the key biomarker compounds that are of medicinal value. So that was a lovely project that we worked on. And we compared then the plants that had been growing inside the tissue culture bottles to those that had been growing in the greenhouse. And when we moved them as well into the greenhouse, we did some metabolite analysis to understand the chemistry. And it was wonderful to see that sometimes the propagated plants that came from tissue culture were actually more biologically active.
Okay, and they kept their activity even after being planted out into the greenhouse.
Yes, and they kept that activity, and they formed the tubers much faster than if you had started it from a cutting. So that tissue culture system actually shortens the lifespan of the plant, and it allows for the plant to become more biochemically mature, if that's one way to actually put that, because the tubers were quite young, but their chemistry was quite sophisticated, and as sophisticated actually as plants that would have been growing for 3 years or 5 years. And these were tubers that were about 6 months old already.
Oh, wow. So that is really accelerating what happens in the plant just by cultivating them basically on their very own, because in this tissue culture systems, as compared to the wild, you will have only that plant and it's a sterile system. So no other organisms included, no microorganisms and nothing.
Yeah, so I think it's the microclimate that seems to be able to control this. And one can change the conditions to shift metabolism towards high production of umkalin, for example, which is one of these key compounds that is thought to have important biological activity.
Do we know anything about umkalin and its function inside the plant? We extract it to use it for medical purposes, but what is it doing in the plant?
I don't think we have any idea, but I would presume that because these compounds have antimicrobial activity, and they are probably also involved in being part of the biological warfare of the plant, assisting the plant from being able to resist pathogens. And so we don't necessarily know their function, but we can draw a few hypotheses with regards to how they might actually be functioning in plants. And just because they belong to these sort of phenolic compounds, which are actually found ubiquitously in plants and are known to have microbial effects, antimicrobial effects in plants in general. So that is the assumption that they might be part of the microbial fighting system, or I suppose even plant immunity, that allows the plant to be able to cope with all kinds of environmental stresses, whether they be biological or even abiotic.
And that is especially fascinating then when you think that when you have the plant in tissue culture, it matures earlier, but it produces also more of these molecules, even though it wouldn't be stressed by other organisms. There are no microbes included. Do you have any hypothesis why this would be? I would just intuitively expect, well, the plant has to defend itself less, so maybe it produces less of the molecules, but you find the opposite.
Yeah, we don't necessarily know why. We think that it might be a control at the environmental level. So, the phytohormones that have changed might be eliciting a new kind of gene expression or upregulating that kind of metabolism. And even though these plants are in a controlled system, somehow they still have a connection inherently have a connection to their life outside of the bottle. It's so strange. Plants can be so fascinating. You can keep them, giving them a summer day every day, giving them everything that they need. But it seems like their genetic constitution can somehow have memory. I'm putting that in inverted commas. And perhaps that is one way also of trying to get ready for a microbial attack. They might be thinking, there are no microorganisms associated with me now, but let me make enough of these metabolites in case I'm actually attacked by pathogens, then I'll already be ready. It's almost as if they get prepared for war, I suppose.
They are proactively careful to survive. That was a very interesting view into the pelargoniums and how you work with them. I mean, domestication of Pelargonium sidoides, how long have people really worked with that?
I'm not really sure in terms of at large commercial scales, but I would presume that 2006, 2007, there were a lot of scientific papers trying to come up with systems of actually how to grow these Pelargonium sidoides on a much larger scale. And since then, there's been a fair amount of foundational information that's actually been built by the scientific community, which has then been exploited in agriculture. And that information there's still parts of that information that's still lacking. We haven't sorted out practices that actually need into this kind of cultivation. But there are farmers within the Western Cape, the Eastern Cape here in South Africa, and I think even in Lesotho, where commercial cultivation of Pelargonium is increasing, and that is increasing to actually meet the commercial demand for the extracts that are made from the tubers of Pelargonium. So at this particular stage, I can't give you numbers in terms of tonnage of cultivated material, but it is definitely a growing industry. and different producers might be growing particular chemo elite lines. And there's a lot of information actually that also lies with individual farmers as they themselves actually test out practices that are most effective for their particular soil types, climatic and geolocations. So it is a growing industry and there is much interest, and it is a very important species in South Africa in terms of commercialized South African medicinal plants.
Mm-hmm. Is it, now this is the Pelargonium sidoides, do you also have such kind of research or programs or cultivations ongoing for other Pelargonium species?
Graveolens, most definitely, because that one has this very important commercialized essential oil. There's a lot of cultivation of that particular species that takes place in more the northern parts of South Africa, that are a little bit more semi-tropical, I suppose. And there's a lot of farming of that particular product. And it's even been grown outside of South Africa and in places like Madagascar and other parts of the world, because it's such an important essential oil. It's used a lot in perfumery, and it has those rosy floral notes. And so that essential oil is very important for the cosmetics industry, actually. Mm-hmm. So it's been cultivated in large masses.
So these two Pelargoniums are the most commonly commercially cultivated species.
Yes.
Yeah, for medical
apart
purpose.
From all the apart from all the pretty Pelargoniums,
yes,
they have literally found themselves all over the world in all kinds of hybrids and cultivars. Those two are the two that are mainly utilized for medicinal purposes, but there's tons of Pelargoniums that are found all over the world, which make spaces really pretty. And anytime I travel outside of South Africa and I see these Pelargoniums, especially in windowsills of apartments, then I get a little bit nostalgic and it reminds me of home. And it just makes me think of how incredible the biodiversity of this country is, and that it is found different homes all over the world.
It has spread its roots throughout the world from when it got exported first.
Yeah.
Thank you very much. Is there anything else that you would like to share? If somebody wanted to know more about the Pelargoniums or about your research, maybe we can hear where we can find you or more information.
I feel I'm really, really easy to find. I'm easy to find on the internet, and I enjoy tweeting. I like to be on Instagram, and often I'll share information about South African plants, and sometimes my escapades to go and find interesting plants in the wild. And so my Twitter handle is @NoxTheLion, and I'm also Instagram @Nox_Makunga. I'm easily to find as well via email, and I can be emailed using Makunga @sun, as in sunshine, .ac.za.
Thank you very much. I think through Instagram, I have also known you. That is also where I got in touch with you. And then we noticed that one of your students had been coming to Umeå and has done his project with me. So that was very, the world is smaller than one thinks.
Yeah, the world is pretty small.
Yeah. It's not just the Pelargoniums that move from South Africa to Sweden and all around. It's also the students. Yeah, thank you very much for this interview. It was really a pleasure having you.
Thanks so much to you. It's been wonderful to be here this afternoon, and I hope you enjoy the rest of your day.
I hope that you have enjoyed this very insightful episode. And while we have still some more episodes on Pelargonium coming up on our podcast, I would love to hear from you what kind of plants you would be interested in to hear about in the future. So maybe you have a favorite plant, or you know a person that is an expert and would be really great to have as a guest on the podcast. Well, visit us at www.flora-l.com and find the contact form on our website to reach out to me and to let me know any kind of wishes for future episodes on this podcast. And with this, I wish you a pleasant day, and I hope to see you back here next Wednesday for our third Pelargonium episode.